Hi, everyone. Thank you so much for coming to Popping the Science Bubble tonight. This is a monthly lecture series that the library has been in partnership with for years. So we're going to hear about some science from some Cal students. Thanks for coming. I'm going to begin with our land acknowledgement statement, and then I'll introduce the program. So, the Berkeley Public Library buildings are built on the territory of the Huichin within the homeland of the Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley throughout the East Bay since time immemorial. The library recognizes that Berkeley residents have and continue to benefit from the use and occupation of unseceded stolen land, and we pledge to recognize the permanence and the rich contributions that the Ohlone people continue to bring to the city of Berkeley and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in our collections and programming. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Louis Trevino and the Berkeley Public Library. So welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussions about a variety of science topics. We have two speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are two graduate students, Kaylin Wang and Daniel Bretthauer. And Annika is our new organizer joining us tonight. This program happens every third Tuesday of the month at 5:30 p.m. at the library and on Zoom. If you're interested in checking out their past seminars, you can visit their website and sign up for the listserv to keep in the loop for upcoming seminars. Also, check out their YouTube page to view this lecture and the scene archive of past lectures. The YouTube page is really incredible. There's a lot of interesting stuff in there, and I highly recommend you check it out. So thank you so much. I'm going to hand this over to our organizers who will introduce our next speaker.
Awesome. Thank you so much, everybody, for coming. So for tonight, we're excited to have Amala John and Eleanor Wang be presenting with us tonight. So this is an open format talk. For those of you who are on Zoom, please use the Q&A function or to ask your questions, and we'll submit that, and we'll ask our presenter that at the end. Or for those of you who are in person, please raise your hand, and our speaker will call on you when there is a good time to answer your question. So to start us off, tonight's first speaker is Amala John, and she grew up in the East Bay, and her fascination with DNA and biology led her to study molecular biology at UCLA. There, she fell in love with plant biology, and she saw how our lives revolved around them and moved across the country to UNC Chapel Hill to get her PhD in plant developmental biology. She then landed back in the East Bay at UC Berkeley as a postdoctoral scholar, studying plant photosynthesis in the Niyogi lab to learn how we can use plants to alleviate climate change. Outside of research, she enjoys exploring new types of dance, gardening, and time with friends. And she'll be telling us all about when plants need shade, the science of photoprotection. So please, take it away.
Thank you so much for that introduction, and thank you so much for having me at this talk. It was really fun to put all of these slides together. Hopefully everyone can hear me. But yes, my name is Amala, and I'm going to be talking about the science of photoprotection. So first, we'll start with this overarching question that I have found myself pondering, which is, why do some plants thrive in full sunlight and other plants prefer shade? Sunflowers are a great example. It's in their name. They require 6 to 8 hours of sun during the day to grow and develop firmly, as opposed to ferns, which typically like shade. They like to be on the bottom of forest floors. And if you switch the environments for both of these plants, they would probably really struggle. And so let's explore one of the reasons why this is the case. So plants need the sun, of course. If they don't get enough sunlight, they tend to starve. They can't make the food that they need to survive. But if they get too much sunlight, they also struggle, and their cells can get damaged, which we'll talk about later. And so there's a sweet spot where they're getting enough sunlight, but it's not overpowering. And they need sun because of photosynthesis. So let's review this very briefly. Maybe you learned this in high school biology, but it's essentially when plants take light from sunlight, as well as water from usually their roots, and carbon dioxide from the atmosphere to convert that into the sugars that they need to grow and develop or store, as well as the byproduct of oxygen, which we rely on for breathing. And it's essential for almost all life. And, thanks to single-celled organisms almost 2 billion years ago that fill the atmosphere with oxygen is one of the reasons why we are here today. Right? So what is it in the plants that actually allows them to absorb the sunlight? And it is chlorophyll. So these are tiny molecules. There's millions of these molecules within the chloroplast of the plant cells. And these are what are actually absorbing energy from light, and then they're using that to drive photosynthesis. I occasionally see these pill forms of chlorophyll. Unfortunately, it's not a very stable molecule, and so you're probably better off just consuming plants instead of trying to consume chlorophyll. But beyond chlorophyll, there's tons of other plant pigments that have a lot of different functions. Some of these are ones that we also study in the NeoV lab at UC Berkeley. Carotenoids also have function in absorbing light energy. And there are anthocyanins and xanthophylls that also have really important functions in protecting plants against sunlight, or anti-inflammatory properties, even functions in reproduction. But chlorophylls tend to be the most abundant, and this is what makes the photosynthetically active tissues of leaves. It gives it their green color because chlorophylls are absorbing red and blue light and reflecting green light, which hits our eyes and then allows us to perceive them as green. And so they're kind of like this original solar panel. And this made me wonder whether solar panels or plants are more efficient at converting sunlight into energy. So I'm going to ask you this question. Who thinks plants are more efficient than solar panels? Raise your hands. Okay, so pretty almost everybody thinks that. What about who thinks solar panels are more efficient than plants? Okay, yeah, we have one person. So it turns out that actually solar panels are quite a bit more efficient than plants at converting sunlight into energy that can be used, which I also found really surprising. So they use something called the photovoltaic effect to convert sunlight into electricity that we use in our homes or in our calculators. Whereas, of course, plants use photosynthesis, but this is at an efficiency of 1 to 6%, depending on the plant, depending on the environment. And the theoretical maximum is 11 to 13%. So we're really not there. There's a lot of room for improvement. And this is a big area of research in studying photosynthesis, ways we can optimize this process. There's a lot of different areas, and I will be talking about one of these areas. And it has to do with understanding how plants respond to too much light. So it's interesting then to think about how humans respond to when they get too much, when we experience too much light. So we also produce a lot of pigments in our skin, such as melanin, that can actually absorb harmful radiation and prevent us from accumulating UV damage in our cells that can, of course, lead to cancer. And when this isn't enough, our skin actually experiences sunburn. And this is also a protective effect. Those highly damaged cells, our body is kind of trying to get rid of it because they might lead to cancer, even if it's really painful. And plants have a lot of different ways to manage too much sunlight. Those chloroplasts that I mentioned, they can actually move around in the cell to try to, have less absorption of light. Plants can droop, so the angle that the sunlight hits them, they can kind of minimize that. And I'll be talking about how plants actually can dispel excess light as heat, kind of as this release valve. So it's really important that plants learn how to manage, plants as well as all organisms really learn how to manage too much light. And this is because of the production of something called reactive oxygen species, or ROS. You can think of them as toxic oxygen. They're kind of unavoidable in like living systems, but there's a lot of different ways that we can manage the production. So if we think about chlorophyll acting as this funnel and taking in sunlight energy and then pushing that into photosynthesis, if there's too much light energy that's coming in at any given time, there's just too much energy that's being absorbed, then can actually go into photosynthesis. So then this starts causing problems. It can lead to the production of more unstable versions of chlorophyll called triple chlorophyll that can lead to the production of these reactive oxygen species. So the plant really wants to minimize how often this is happening, has lots of different ways to prevent this, formation of different pigments that manage can scavenge for these as well. And what I'm going to be talking about is how instead of waiting for these to be produced and dealing with it, there's another method that can actually dissipate this and prevent it from occurring as a release valve. And this is releasing light energy as heat. And so I think of this process kind of like a surge protector. It's called non-photochemical switching. And if we think about your home's electrical system, every once in a while, you actually might experience surges of energy that come in. This can happen after a power outage or during like a lightning storm, which is why it can be good to like unplug large appliances during a power outage. And what's happening is that there's too much energy that's coming into your system, and this can hit your appliances and damage them. This actually happened to me a lot in the last apartment I lived in because had a really old electrical system, and my appliances kept breaking. And so I invested in some high quality surge protectors. And what these do is that they basically divert this surge of energy away from your appliances and it grounds them. And I think that this non-photochemical quenching, or NPQ, does a similar thing where the chlorophyll can get excited, and this energy from chlorophyll absorbing light energy can be used for photosynthesis, which is also called photochemical quenching. And it can also, of course, as we mentioned, it can lead to energy buildup that can lead to toxic oxygen, reactive oxygen species. And then it has this other method of non-photochemical quenching to release that light energy as heat. And so plants that are in very bright conditions are usually releasing small amounts of heat. You might not be able to perceive that happening. So yeah, let's talk about now how NPQ changes based off of different light conditions. So in dark conditions, photosynthesis is low and NPQ is pretty low. And then in conditions of very high light, photosynthesis can be saturated. And this is where NPQ is really, really active. If you can imagine, if photosynthesis, or if NP2 is active and it's releasing that light energy, you can imagine then that there's less energy available for photosynthesis. So in that way, it indirectly inhibits photosynthesis from happening. It's kind of like if you have a surge protector that's constantly diverting energy from your appliances, you won't be able to use those appliances. Similarly, photosynthesis is pretty much not going to be full potential when NPQ is active. And this tends to be very important when light conditions are fluctuating a lot. So on a normal day, you're going to have maybe bright conditions for maybe a couple minutes, and then clouds might come in and move out and come in. And plants have to be responsive to these changes in their environment, depending on the plant. And so plants act depending on the plant, but a lot of plants actually tend to have very optimal photosynthesis in low to moderate light, not when they're in like blazing sun. But when the cloud cover goes away, then NPQ becomes activated and vice versa. Now, this can become an issue if there is a lag in NPQ becoming activated and released. So what that kind of looks like, if we look at this chart, if these blocks are showing us sunlight over the course of the day, which is going to fluctuate, and the red line is NPQ that is trying to match what's going on. So say we enter highlight conditions, NPQ is going to be turned on to protect the plant, and then the light levels drop again. And so NPQ is once again decreasing, but there's a little bit of a lag here. And so this lag is basically wasted potential where we could have been photosynthesizing, but we're not. And maybe for a couple minutes, that's fine, but you can imagine over a day, over weeks, over months, over the life of the plant, this can lead to a lot of waste and efficiency, and might account for those low efficiencies, or one of the reasons for the lower efficiencies. It's estimated that 10 to 20% of their potential for photosynthesis could be wasted from this delay in NPQ relaxation. And what we might prefer, or what some really optimized plants might have, is a very small delay where the sunlight that is increasing leads to very sensitive changes in photoprotection. And you can imagine that a lot of different types of plants experience different NPQ dynamics. So this is a chart made by, with data accumulated by a previous grad student in the lab, who took a ton of different types of plants and actually measured the rate of induction of NPQ and relaxation and took the ratio of that. So on the left side, we're seeing plants that are very sensitive, that can induce and relax very quickly. And on the right down here, we're seeing plants that are not as sensitive. But that's not showing us the level. So these can still have very high levels of NPQ, but they might just not be changing very quickly. So plants like ferns, which tend to be in shaded conditions a lot, they tend to be really sensitive because they're getting mostly shade, but they might be getting flecks of sun coming in and out. And so they need to be very sensitive so they're not getting damaged. As opposed to plants like these, which tend to be in tropical or desert conditions where they're anticipating very bright, sunny conditions pretty constantly. And so they kind of just like keep their NPQ at higher levels, and they're not really expecting to decrease. And so thinking about the different types of plants that are more or less optimized in our lab, we also think about optimizing NPQ dynamics in different crop species. Crops are growing in like pretty unnatural environments in like monoculture, and they might have been domesticated considering yields or how easy it might be to grow them on a farm, but not necessarily for optimal photosynthesis. And so we after decades of research, it was found that these three genes are really important for improving the dynamics of NPQ. So PS, BS, VDE, and ZEP, it was found that if you can increase the levels of these three proteins, you can actually have more rapid dynamics, which is what we want. And this work was done in soybean and tobacco. And they found, so on the bottom here, we're showing NPQ dynamics in a wild type or like a normal plant that we would compare it to in the dark green and gray. And in the lighter green shade, as well as this red shade, we're seeing plants that have elevated levels of these three proteins. And we find that NPQ levels are decreased. And accordingly, if we look at one of the outputs of photosynthesis, which is showing carbon assimilation levels up here, we see that these levels are also increasing, suggesting that photosynthesis is occurring in more optimal ways. And so together, this work is a tool to help plants sequester more carbon from the atmosphere and into the ground, and also just to grow a lot more efficiently. Things that a lot of other plants are already doing, but we can try to increase our knowledge of these processes. And so going back to our initial question, yeah.
Can I ask you a quick question about the previous thing?
Yeah.
The lower levels of NPQ, should I read that as like more sensitive or quicker reacting, or just kind of shift is lower across the board?
Yeah, that's actually a really good question. So this, these data I'm showing here are specifically in fluctuating light conditions. So we're going from, high light to low light to highlight a lot. And so in those conditions, generally NPQ is decreasing. We're not turning it off. We don't want to do that because NPQ is really important for protecting the plant. But essentially we know that NPQ is still occurring, but we're seeing less of it that is lacking essentially. Yeah. Okay, yeah, back to our initial question. Why do different plants require different amounts of light? There are actually more explanations for this than just, what I can talk about in, this like 15 to 20-minute talk. But our general takeaways are that how much and how fast NPQ can be induced generally correlates with light tolerance. So plants like ferns that prefer the shade tend to have like pretty quick responses, but they might not have as much NPQ as plants that are growing in the desert or used to really high light conditions. So full plants tend to have really, really strong responses, but maybe they're not being as sensitive. And another thing that is really important to consider is the trade-off between photoprotection and photosynthesis. Generally, it seems that there will be slower growth with plants that sustain really high levels of NPQ. And we're interested in exploring the different kind of plants like ferns that tend to be really sensitive. I study sorghum and wheat, and we're trying to optimize NPQ dynamics in these crop plants. And with that, I'd love to thank my lab, as well as my advisor and our lab manager for all of the support that they have given me. And for this presentation specifically, my mom helped me run through my slides, and I've had conversations with friends who helped me know what I should lean into and not. So, and yeah, with that, I can thank you for listening and being here, and I can take any questions if I can answer now, or I can look up later for you.
I wonder for the same plant, does the NPQ switch or the on and off lags change at all if the plant is in winter or summer, just because of the amount of daylight available?
Oh, definitely. Yeah, I think so acclimation to different light conditions definitely impacts like the rate and level of NPQ. So if you take a winter plant or like a plant that's acclimated for lower light conditions and you throw that into high light conditions, it's going to struggle a lot more than a plant that is like slowly acclimating to high conditions, which is another thing. I didn't really mention a lot about climate change, but because we're having more unpredictable weather events, our plants are going to probably struggle a little bit more to respond to really unpredictable environments. Yeah.
I was also wondering, so the efficiency of photosynthesis seems really low based off of what you said. So is there like some sort of trade-off that's going for that, like because it has a lower efficiency, it's just better, it's more resilient, or like why?
Yeah, that's a good question as well. I think something I forgot to mention is like, why is it that plants are going to be very different than these machines, these solar panels, right? Because they're living organisms and they haven't really evolved for efficiency, they've evolved for survival. They're trying to they're trying to reproduce, produce seeds and have like the next generation of, plants. And so they're doing a lot of things. They're photosynthesizing, they're protecting themselves from pests and viruses. They're, like I said, they're reproducing. So they have a lot of things going on. And so they're really just, photosynthesizing enough so that they can survive. Your question was specifically about trade-offs. But yeah, I think that's kind of like how I think about that. Like they're really optimized for survival. And that's probably the most important trade-off.
Thank you.
Yeah.
Kind of related to that, in the study where they turned down the NPQ and they saw more efficient photosynthesis, did they see other like side effects of that in the
Yeah.
plants?
Yeah, so those studies, that they found that if you can, and actually it was, I guess that was slightly misleading because it does show decrease in NPQ, but really what's happening is that the rate of NPQ is, of induction and relaxation is optimized, so it's occurring faster. And so yeah, that does mean overall you have less NPQ, but basically the NPQ is there when you need it and not there when you don't need it. And they found that this was able to increase yield pretty significantly in those two experiments. But there are other experiments where they tried this similar experiments and they found that that wasn't able to improve growth or yield. When I say yield, I mean like how much biomass you can have per like given land and like number of plants. What else did they see? They also saw improved, there were other studies that show that water use efficiency also increases when you can improve NPQ dynamics, which is also improving photosynthetic efficiency. They also, yeah, and probably the most important thing is that carbon assimilation, so the output of photosynthesis is also increasing in efficiency. Yeah, those are kind of the main, like the big kind of findings that they have. Yeah.
It does not appear that we have any questions on Zoom in the Q&A and the chat.
Right, let's give Amala one more round of applause. Switching over to the next. All right.
I'll mute the mic. There isn't a way to tell, so.
Pleasure to introduce our second speaker of the night, Eleanor Wang. Eleanor is a fifth-year PhD student studying microbiology at UC Berkeley, as well as an artist, zine maker, and the art director of the Berkeley Science Review. They currently study how a strange group of microbes known as archaea make and use vitamin B12 and similar nutrients that have worked also in various biology research areas before starting their PhD. They like to climb rocks, touch sea creatures, read books in uncomfortable sizes, and are learning to play the electric bass. If you're ready.
Hopefully everyone can hear me all right. Thank you so much for being here. Thank you so much for the lovely introduction. Yeah, today my talk will be about microbes. I work on a weird group of microbes known as archaea, but the format of my talk today will be slightly unconventional in the sense that it comes in the form of a zine reading. And if anyone is rolling in late, I can still hand these over so everyone can pass around my zines. And if anyone is on Zoom and wants a copy of the zine towards the end of my presentation, I have the free PDF available. So look out for that. And yeah, so today the title of my talk is "It's a Microbial World After All." The littlest guys truly do the most. And as someone who is 5'1", I think that when I first started learning about different life forms, I felt some affinity towards microbes for some reason. But before I get into the zine reading itself, Nice to it. Yes. A little bit about me. I went to UC San Diego for my undergrad, where I first learned about genes and developed a love for microbes. And so these two things kind of happened because I was really involved with a lot of different science communication type of efforts. And I feel like being in spaces where people are really curious about different kinds of science has always been some of my favorite spaces to be in, because I end up learning a lot about things that I might not have necessarily known about before knew that I wanted to know about. And so that's how I first kind of started getting into microbiology. I wanted to thank my dear friend Aria, who was part of that journey and who also made her Zine Fest debut around the same time that I did. And so today my presentation will also in part be an advertisement for the concept of the zine. So I would highly recommend that when your local zine fests happen, to please go check them out. I believe Berkeley Public Library also at many branches has free zines available. And yeah, I wanted to also express my deepest gratitude to the incredible zine community for being such a powerful source of love, joy, care, and revolutionary storytelling. And so hopefully I can make them proud today. Yeah, so hopefully everyone has a copy of the zine that I handed out. I tried my best to make the slides match what's happening in the zine. The zine itself, it's the format of a classic mini zine, but if you unfold it, I also included some like internal contents. In terms of what I'll be going through in my slides, it'll mostly just be outside contents, and then I've included the inside as additional resources for anyone who's interested in learning more. Cool. And so without further ado, if we flip to the first page, I wanted to start by defining what a microbe is, because we typically aren't really thinking about them as macrobes or macroorganisms. There's a thing in my screen. So yeah, microbes, also known as microorganisms or microscopic organisms, are creatures that in essence are too small to be seen with the naked eye, meaning that you need a microscope to see them. And so that's a pretty broad, wide encompassing definition. But if you think about viruses, so coronaviruses or like other viruses that sometimes infect other microbes, so like bacteriophage, that's included. Same with a bunch of different fungi. Same with archaea, which is the group of microbes that I work on, and bacteria, protists, and other funky friends are also within this definition. And so on the bottom panel of this first slide, I wanted to also say that microbes live in important places, i.e. everywhere, but I feel like the field of microbiology generally focuses on some key areas, including bodies of water, so like rivers, oceans, lakes, things like that, the soil, in and on other creatures, and that's my way of saying what the microbiology field colloquially knows as like host associated. So like think about maybe like the guts of an insect or our guts, or like the skin microbiome and things like that. And they also exist in fermented foods. Some people also study things like the industrial microbiome, but it's kind of hard to categorize everywhere. So yeah. And if we flip to the next page of the zine, microbes also live in community with us, with each other, with the world at large. And I think that's something that also really appealed to me when I first started learning about microbes, like the fact that you can't see them, but that the way that they shape the world is by interacting with the things around them. And so if we think about actually the estimated biomass distribution on Earth as measured in like gigatons of carbon, so just if you weighed every living thing on Earth, there's this funky little graph here. You might not have seen one shaped like this before, but really what it represents is like a pie chart, but in square form, and you sliced the square into a bunch of different shapes that should be the same size as what data segment they represent. So animals actually represent only 2 gigatons out of, I think the entire square is over 500 gigatons or so, whereas bacteria are estimated to be about 70 gigatons. Archaea are 7, fungi are 12, and plants are most of the world, it seems. So yeah, if you think about the fact that microbial cells are so small that you can't see them, if you compare this numbers wise to the number of animals that are in the world, microbes are very small, but they are very many. So yeah. And so in the communities that microbes live in, they will interact with each other. They will interact with the creatures that they live on or in, known as their hosts, and they will also interact with their environment. I will say that as a zine maker, I specifically chose to choose some more obscure artistic images, but hopefully nothing is too strange and confusing. And these images are also not to scale, but yeah, these interactions within the microbial communities can have positive, neutral, or negative effects to varying degrees and come in very many different forms. And so microbes can, for instance, hurt their hosts. So if we think about plant disease, human disease, I've included an image of the coronavirus SARS-CoV-2. They can help their hosts. So if you think about fixing nitrogen, if you've ever learned about like root nodules in plants that fix nitrogen so that the plant can use that nitrogen to grow, those root nodules are formed by a group of bacteria. And we also, as people, take probiotics to help improve our gut health. Microbes can help each other by sharing nutrients with each other and also doing a bunch of other stuff. They also can hurt each other. So there's predation that happens in microbial communities, and they also produce antibiotics to kill other microbes often. And there are also many things that I'm not listing here, but feel free to ask me questions about them. I will do my best to answer. Yeah. All right. And then, oh, that was on the second to last page, I guess. But finally, this is the closest to the area that I work on, but microbes also make and eat a lot of very important chemicals, i.e. they cycle the elements of life. And so by that I mean, if you think about the periodic table and the number of chemicals that make up life, so like carbon, nitrogen, phosphorus, oxygen, microbes are really essential for things like the carbon cycle, for nitrogen cycling, going back to the example of nitrogen fixation and root nodules that I just mentioned. They will cycle phosphorus, sulfur, like trace metals and minerals and other really important components of life. And then I've also included a couple of fun facts in the zine. So one is that the first bacterium, so not just like chemicals of life, but microbes evolve really quickly. So a lot of the time, their enzymes are good at evolving to eat things that people might take a little bit longer to learn how to eat. And so, for instance, the first bacterium to eat plastic, named Bidionella sakaiensis, was discovered by Japanese scientists in 2016. I've used the nitrogen fixation example already, but this bottom one over here was, I think, one of the key pieces of information that I learned early on that really convinced me that microbes were way more important than anyone gave them credit for, but which is the idea that cyanobacteria, who were the originators of photosynthesis, so this is very topical today, are responsible for the great oxidation event. And so the event where Earth's atmosphere became filled with oxygen, and then everything had to adapt to learn how to process oxygen. And these cyanobacteria, or just single-celled algae, still do about half of the photosynthesis on our planet. And yeah, so in essence, by transforming the fundamental building blocks of life, I'm reading off my screen, microbes shape our world despite their unassuming size. Yes. And if you flip to the very back of the zine, I have a few. To the left, I have journaling prompts. And so these are reflections about things that you can maybe cannot see without your eyes, but make an impact with your life. I won't read them off the board. I have an About Me section, and then I also have a QR code for the PDF of the zine. Yeah, and I'll leave it on for a second to take questions. There's still like a good couple of minutes before I jump into talking about what's on the top of the zine. All right, everyone has a copy of the zine anyway, so, all right. Cool. And the back of the zine is mostly text, but I would describe it as primarily like a very rough overview of what I work on as a PhD student. So if you go section by section, so like from the screen, yeah, if you go directionally from the left half down to the bottom and then the right half down to the bottom, these are the different components of the contents of the back of the zine. And so, yeah, I work on archaea and how they make a group of vitamins known as coronoids that includes B12 and how those processes end up shaping the way they interact with other microbes in their communities. And for the most part, what I do on a day-to-day basis is I do a lot of coding and then I do a lot of growing things and then measuring what is growing and how well they're growing or whether there's something in it after you grow it. So yeah. And I also included a bunch of resources for anyone who is interested in learning more about microbes in general. There's one of my favorite books is by Yang, who is actually an Oakland-based science writer, who wrote like a pretty well-selling book called I Contain Multitudes. It's mostly about gut microbes, but I think it's really well written. There is a really fantastic 12-minute video by Dr. Dipti Nayak, who is a professor at UC Berkeley, about archaea in general and the tree of life. And if you are interested in watching fun videos of microbes under the microscope, I also really like Journey to the Microbe Cosmos on YouTube. A lot of the videos, I believe, are narrated actually by Hank Green. And if you also want to support local science storytelling, I think you should continue to show up for Popping the Science Bubble. And there's also Berkeley Science Review. That's a grad student run magazine that I have, I guess, a conflict of interest in plugging. But if you're looking for something else that you think I might be able to help with, please reach out. And I will put this back up. I also will have free copies of another zine that I have about the ocean microbiome available. I think I should have enough copies for everyone today, but the PDF is also available for free. And if there are any scientists in the room, someone I know is looking to collaborate, like create collaborations between scientists and art students. The application deadline is in about 5 days for that. The goal is to kind of make science comics together. And I also have a bunch of copies of the Berkeley Science Review to give out. And yeah, without further ado, I think that is a wrap there and take any questions.
So, on the inside of your zine, you say that archaea known as methanogens
Yeah.
are responsible for about 80% of global methane emissions every year, which is really striking.
Yeah.
So, is that a mechanism through which, and not through like human-induced emissions can occur, or is that something that's happening completely separate? And then when we talk about the methane emissions that we're thinking about changing year to year, a lot of that is coming from like our activities that's totally separate from what the archaea are doing, if that makes sense.
So I think, so to repeat the question, my understanding of the question is that basically like for methanogens that are a group of archaea that make methane, I wrote in my zine that 80% of them, or they're responsible for 80% of the methane emissions on Earth. And so this does include anthropogenic related activity. And so, for instance, in landfills, which is like a pretty big source of methane emissions, what happens a lot of the times when you put out food waste, I believe, is that first it gets fermented and processed by a bunch of bacteria, and then the archaea are then able to turn a lot of those chemicals into methane. And so, does that kind of answer your question?
totally what my question was. Thank you.
Amazing, thank you.
I would love to learn a little bit more about what exactly makes the archaea so different. My high school biology was, all right, you have the prokaryotes and the eukaryotes and the nucleus, and that's it. So I don't know that much about the archaea. What's different about it?
So the question is what makes archaea so different from bacteria? And if anyone has taken intro biology, as Daniel said, you're usually introduced to the idea of like, oh, well, you have the prokaryotes and they're little guys, they don't have a nucleus, they're simple, like they're not, and then you have the eukaryotes, so like animals and plants and fungi. And up until, so this is kind of like the first section of the back of the zine, but in about 1977 when this idea that archaea are actually really different from bacteria was first introduced. And this idea was based on like a sequencing approach where they sequenced the genome or a 16S like ribosomal, like this part of the gene, and then they were like, okay, let's use this sequence that all life forms in life have and kind of compare them to each other across archaea, bacteria, and eukaryotes and see how different that sequence is. And by using like that degree of similarity, we can tell how different these organisms are from each other evolutionarily. And this was kind of first when they found that, wait, actually, archaea have a really different sequence compared to bacteria. And it seems like this sequence is as different from bacteria as bacteria are from eukaryotes. And so this was quite contentious back in 1977 when it was first introduced, but and then around the 90s was when it kind of became more commonly accepted. It became more integrated into textbooks and things. But even then, like I think a lot of the main differences between archaea and bacteria have to do with some of their cell biology. And for instance, the methanogens that we mentioned earlier, the only organisms that rely on that process of making methane as their central way of making energy are archaea. And so because we've spent so long of our, I guess, Western scientific history thinking of archaea as basically the same as bacteria, but just a lot harder to grow, I think that there's just
fungi.
Fungi? Oh, fungi are eukaryotes. Yeah.
Very often.
Yeah, they're eukaryotes. But
What's a eukaryote?
so a eukaryote is an organism that has something called a nucleus. And so for instance, humans are eukaryotes and plants are eukaryotes, but bacteria and archaea are like prokaryotes in that they grow as single cells and they're like cellular compartments, just don't have the same structures that eukaryotes do.
They have DNA.
Yes, every living thing has DNA. Yeah. Yes. Yeah. Thank you for the question.
Yeah, I was wondering if the vitamin D in different organisms are slightly different.
Oh, B12 or the core.
Sorry, vitamin B12.
There's so many different vitamins.
Yeah.
And they just have a letter and a number.
Right, and like also B12 is made, or the supplements are made in microbes.
Yes. So commercially, so our vitamin supplements for B12 ultimately all come from bacteria. So they have big plants where they grow these bacteria in like commercially, they like engineer them so that they make a really high quantity of B12. And then they extract it all and then they sell it to you as a pill. And then yes, different microbes are able to make different versions of coronoids. So B12 is an example of a coronoid. And there's like, I think about 17 or 18 naturally occurring coronoids and different.
They're B12 or generic B12?
Sorry?
Just generic B12 or any particular kind of special B12? I know there's a few of that, like
So-
the supermarket, they have like a bunch of.
People can only
Cobalamin or.
process B12. Yeah, cobalamin is B12.
I would read it.
Yeah. Yeah, so cobalamin is B12, but B12 is one out of those like 17 or 18 different kinds of coronoids.
Block it.
Oh, sorry.
Fine, right.
But yeah, And different microbes can have different preferences for out of those quarterlies, which one's their favorite. So, yeah.
Any more questions? If not, then let's thank both of our speakers one more time.
Thank you so much. Bubble. We will be returning on February 17th. I think tentatively at the South Branch this time. Or
Central.
And still, so right here again.
Yeah.
We're pretty much done with our tour.
Yes.
And so you can sign up to get our email notifications using this handy dandy QR code that we have over here. Or by signing up using bit.ly/PTSD emails. And I think we have some of the topics for our next.
Yes. So for February, the 17th, Tuesday, we will have two speakers. One from neuroscience. We'll be talking about psychedelic drugs and how they reopen critical periods. And we have a second talk in vision science about visual processing and the limits to acuity. So very exciting.
So we hope to see you again in just about a month's time. Thank you so much for coming out again.
Thank you.